mention of the word mutation conjures up images of bizarre lab experiments with monstrous results. Science run amok. Of course, these are the products of mass entertainment and not scientific understanding. What we do know is that mutations are inheritable changes in genetic material, alterations in the DNA code. They may be induced by environmental or man-made agents. They may affect the phenotype of the affected organism or its offspring. And perhaps most surprising, especially for those influenced by the science fiction tradition, not all mutations are dangerous, nor damaging. In fact, some mutations provide advantageous characteristics and are, ultimately, desirable. In this program, you will learn how a mutation in the DNA of a cell can provide a source of genetic variability. You will also observe a back mutation simulation, which is part of a test to determine the mutagenic potential of various chemicals. You will also learn how certain human activities affect the rate of mutation of DNA. By the end of the program, you will appreciate the scope and significance of current research on certain DNA technologies and some of the implications of patenting genes. If we were to uncoil the DNA in a single human cell, it would measure 1.5 meters long. Now, we have trillions of cells in our bodies, so you can imagine how many meters DNA that is.
We probably need all that DNA in a cell to store all the genetic information that makes us who we are.
It's amazing to think that DNA contains the instructions to make proteins, which are part of our hair, skin, tissue.
And it's even more amazing to think that all living organisms have the same basic molecule, yet the instructions in the DNA of a plant cell produces such different results than the instructions in an animal cell.
The DNA molecules are made up of the same genetic building blocks in both plants and animals. That's 4 different nitrogen bases, a sugar, and a phosphate. The ultimate source of genetic variation and overall diversity of organisms comes from mutations. Mutations rearrange those genetic building blocks. Probably the most profound effect of mutations is not immediate, but occurs over the long term within a population rather than individuals. In sexually reproducing organisms, individuals carrying a mutated gene which is recessive must combine with other individuals carrying the identical mutated gene in order for the mutation to appear in the progeny. Since the number of possible combinations in sexually reproducing organisms is 2 to the power of n in the gametes, 2 to the power of n squared in the zygote, it takes many generations for the mutation to become viable in a population. Some scientists believe that when the genetic code is altered by changes in the DNA, that those changes are what produce the variety of organisms. We're going to deal with specifics of how a certain species has evolved through mutations. We will look at some of the smaller changes. And one of those smaller changes is the phenotype expression due to mutation. So, do either of you know what mutations are?
Mutations are changes in genes.
The word mutant has kind of a negative connotation, but not all mutations are negative, are they?
Mutations are changes in the DNA that affect gene expression. Now, some of these changes are completely inconsequential because the DNA is paired, or there's a section of the DNA that's inactive, or there's a duplicate strand of DNA that can code for the protein. Mutations can be anything from harmless to lethal, and we'll look at some of those examples of mutations later. First, let's review the genetic code.
Every gene in a chromosome has information to produce a protein.
And DNA is like a recipe for those proteins.
Right. Sequence of the nitrogen bases in a DNA molecule determines the sequence of amino acids in a protein. Remember, RNA polymerase enzymes read the appropriate section of DNA and then build a messenger RNA strand as a result. And that brings us to the triplet code. Do either of you remember what a codon is?
A codon is the messenger RNA sequence for 3 nitrogen bases that code for specific amino acids.
And with 4 different nitrogen bases, there are 64 possible combinations of 3 nitrogen bases, which make up codons, and code for specific amino acids.
Right, because 4 to the power of 3, or 4 cubed, is 64. But there are only 20 amino acids, so that means some codons code for the same amino acid.
And they can also signal the beginning or the end of this translation.
Right. Let's build a short 9 amino acid protein. And we'll use this DNA here. Actually, Peter, can you put these last 2 on? Thanks. As our code. What would be the first step?
Well, the first thing that we'd have to do would be transcription. And for that, we need another mRNA strand, which would match up the codes on the DNA strand. Like this. And on the end, we would have a terminator codon.
In this mRNA strand, specific transfer RNA brings specific amino acids to the ribosome, and a specific amino acid chain is built.
Kasha, why don't you line up the codons with the amino acid?
Okay, they would all match up like this.
Okay, good. And as you said before, AUG is an initiator codon and UAA is a terminator codon. Let's see what happens when we replace the bases in the DNA molecule. That's called a base substitution mutation. Okay, take a look at the codon for arginine and look at the corresponding DNA segment.
GCA?
Right. Now replace this C with that A.
Okay.
What happens to the codon now?
Well, it should have another U.
There you go. Okay, if that change were made in the DNA, would we get the same protein?
No, because the codon would now code for leucine instead of arginine.
Right. What we've put out here is a sequence of amino acids. The sequence of amino acids is important to proteins. And we've put them out in a line here, but each of these amino acids is a 3-dimensional structure. And the sequence of those 3-dimensional structures determines the overall shape of a protein. The shape that a protein assumes determines its function. Now, there's another 9 amino acid protein that we can look at. The first one that we made here was called ADH. And what I'm putting out here now, actually, can you use the leucine from that other mutation?
You bet.
Right. Okay, this is a molecule of ADH. It's 9 amino acids long. This is a molecule of oxytocin. It's also 9 molecules long. And the only difference between these molecules, we saw in the leucine, the 1-base substitution, and isoleucine instead of phenylalanine. Now, both these molecules have very different functions. They have very different shapes.
Okay, so if we change the sequence of the amino acids, then a different protein is produced, which has a different shape, which would mean that it also has a different function than the original protein without the mutation.
Absolutely. There's another kind of mutation, and it involves the addition or deletion of 1 or more nitrogen bases in the DNA sequence. Let's take a blank piece of paper and let's delete this G from the DNA. How would that affect our protein?
Well, it would cause a shift in the way that the DNA is read. So that means that the C would go with the 2 A's, and because it needs to be a triplet, and the other letters would change too.
Right. The ribosomes simply read the information in groups of 3 nitrogen bases. So the addition or deletion of a base means that there's a shift in the frame of the 3 bases. That's why it's called a frame shift mutation. Now, this can also happen when a piece of DNA becomes separated from the chain. X-rays can make that happen. If pair enzymes don't splice that DNA back in the appropriate spot, then there are dramatic changes to the genetic code.
I can see how frame shift mutations could have such a major effect. I mean, they create a completely different protein. But Base substitution doesn't seem to be all that dramatic. It just causes a single amino acid change.
Still have lethal effects. Have you ever heard of sickle cell anemia?
Oh, that's when you have a normal red blood cell, and then some of the cells shaped like sickles.
Right. That change in shape is caused by a single base substitution mutation. A thymine is replaced by an adenine in the triplet that codes for the 6th amino acid of the beta chain of the hemoglobin molecule. That single base substitution completely distorts the shape of the red blood cell and makes it much more prone to clotting. There are lots of other serious side effects as well.
So the original DNA triplet would be CTC, and the mutated DNA would be CAC.
Okay, so the triplet CTC coded for the mRNA GAG, which in turn codes for the amino acid glutamate. The mutated triplet CAC, which codes for the mRNA GUG, which creates valine.
Right. Sickle cell anemia can be lethal when a person is homozygous for the sickling allele. The replacement of glutamate by valine causes red blood cells to assume an abnormal shape. This sickling of red blood cells in homozygous individuals leads to obstruction of blood flow in capillary beds and small vessels. Poor oxygen and nutrient transport, and consequently poor tissue development, are the result. In turn, these factors may result in bone, lung, or intestinal damage, kidney failure, brain damage, and even heart attacks. These are pleiotropic effects, when one gene affects many characteristics. Damage to that gene results in many symptoms.
There is really a heterozygote advantage, and that situation we know has occurred in things such as sickle cell anemia, where if you go to Africa you find a high ratio of individuals with sickle cell anemia. Carriers of sickle cell anemia, in fact, have some protection from malaria. So what you need is two things. One, you need some individuals in a population that carry a gene, and you need some environmental selective criteria that makes those individuals more viable than those that don't carry the gene.
Are there any other known mutations in human genes that can provide protection?
Actually, there are, Peter. In the 1970s, an Italian doctor discovered that some villagers in a small Italian village carried a mutant gene that allowed them to produce a lipoprotein, which protected them from heart disease.
That's a mutation I wouldn't mind having.
That's what the Italian doctor thought. So he isolated the gene, cloned it, and patented it.
Patent a gene? Can you do that?
According to the law, you can. Patent law applies to new life forms created by genetic engineering.
In this case, the mutation occurred naturally. So wouldn't that be considered a product of nature, and then it wouldn't be patentable?
Well, traditionally, industrial patent regime wouldn't allow patenting of a naturally occurring substance, but a new interpretation has led to gene patenting. If a gene is isolated and then substantially altered, it can be patented.
The mutant allele carried by the Italian villagers has the potential of being very valuable. So who get the economic benefits?
That's a good question, Peter. Technically, whoever owns the patent has the right to any profits from it. But it's a very controversial subject.
What constitutes a biotechnical invention that can be patented?
Firstly, it must be a novel item. That is, it can't have existed in prior art, prior inventions beforehand. Secondly, it can't be obvious. That's obvious to someone who's skilled in the art of in this area. And thirdly, it must have utility. It can't just be of academic interest. This is just a neat thing that I've found. It must have some actual utility, and that must be illustrated within the patent application.
So who's applying for these patents?
Well, researchers are certainly interested in obtaining patents for their inventions, and biotechnology companies are very keen to have their inventions patented because it protects their interests.
What are the implications of patenting a biotechnical product from a legal and social point of view?
This gives rise to a lot of concerns that have been expressed, in particular from the environmental communities and the animal rights communities. Recently, we've seen a coalition in the U.S. of anti-biotechnology activists and religious leaders who oppose patenting of living organisms. And the concern seems to focus on making living organisms into commodities. And what that means from an ethical point of view, many people have grave concerns about this.
Once a mutation takes place, all the descendants of that organism will carry the mutant trait, if mutation is germinal. Germinal cells are the sex cells. Germinal mutations are transmitted from parent to offspring if the mutation occurs in the eggs or sperm. Somatic mutations, which occur in the other cells of the body, only affect the individual that has the mutation, not the offspring.
So what's this test called anyway?
Called the Ames Test, and it's used to determine the mutagenic strength of various chemicals. We test the mutagenic potential in bacteria to help us determine if a substance is mutagenic in animals.
Could we use any kind of bacteria?
The bacteria we use is a mutant strain of Salmonella. It can't produce the amino acid histidine.
So Salmonella can't produce histidine, but without histidine it would die?
Exactly. In this test, we test the potential mutagenic agent to see if it can back mutate the Salmonella so that it can produce histidine. We mix the potential mutagenic agent with Salmonella, which has already been mixed with rat liver extract, plate it onto a histidine-free medium, and then incubate it. We're also going to have a control group. So, Kasia, could you label a sample and a control? Thank you. Okay, the first part of this test is to put some of the bacteria into the test tube. Now, only into the sample test tube will add potential mutagen. Control, we will leave just with the bacteria and rat liver extract. Good. Now just give that test tube a little gentle shake to mix up the bacteria and the substance. Good. Okay, now Peter, would you please plate the bacteria onto this histidine-free medium? Just gently shake the plate and that'll mix it around better. Good. Okay, you need to do the same with the control. Excellent. Now it's a waiting game. If we were doing this for real, we'd incubate these two plates for 2 days at 37 degrees Celsius. And these are the results that we would expect to see.
Well, our sample has more bacterial growth than our control.
And what does that mean, Kasia?
Well, that would mean that our mutagen mutated the Salmonella so that it would produce histidine again and therefore survive.
Right. At the beginning of the test, the Salmonella bacteria are spread over the surface of a growth medium that lacks histidine but contains the chemical that's being tested. If any bacteria grow, it means they have mutated. They've gone from the inability to produce histidine to the ability to produce it, and thus survive. This is known as a back mutation, and the chemical being tested is called a mutagen.
We can't just assume that because a chemical is tested as a mutagen in bacteria, that it will be a mutagen in animals.
No, but what we do know is that 90% of the substances that test positive in the Ames test are mutagenic in animals, and that non-mutagenic agents in the Ames test are generally non-mutagenic in animals. It's not 100% reliable, but it is a good indicator of the mutagenic potential of various chemicals.
In our own laboratory, we've done an Ames test simulation, and I was wondering how the Ames test compares to the Mutatox test. in testing for mutagens in the environment?
Quite similar. The Mutations test was designed on the basis of the Ames test. However, the only thing that's different is we use a marine bacterium that is found commonly in the ocean. So in a normal wild-type state, the bacteria produces light. However, with Mutations, we're using bacteria that does not produce light or produces very little light. And when exposed to certain toxicants or certain mutagens, the mutations within the bacteria actually cause the light to come back on. So in that sense, the Mutations is very similar to Ames.
There's a great deal of research that's been conducted over the past 10 to 20 years trying to identify a variety of carcinogens that exist in our environment and that can cause cancer in humans. Only a small number have been proven to function in this way, but those that have, we know, are really quite potent in this capacity. A couple of examples that are known to be very active carcinogens would be some of the chemicals, for example, that are present in cigarette smoke. Sunlight, especially the ultraviolet rays, are very effective as carcinogens in causing various forms of skin cancer. And there are also a variety of chemical substances, either derived as industrial byproducts or in some cases as products of radioactive fallout or nuclear testing, that have also been identified as very potent human carcinogens. But probably one of the most active is cigarette smoke itself. The basic idea that has emerged from these studies is that if we could identify all of the environmental carcinogens, and in particular the half dozen or so that are known to be especially active, and somehow eliminate them from our environment, either by cleaning up the environment or changing people's habits so that they stop smoking, use sunscreen, and so on, that we could probably reduce the overall incidence of cancer in North American society by something on the order of 50%. In other words, of all the people who are getting cancer now, half of those, in theory, are preventable by changes in our environment and lifestyles. The basic idea is this. Our cells, even in a normal, healthy person, have the capacity to divide and differentiate, for example, in response to wounding or damage to tissues. Cells need to divide to repair those wounds, form scar tissue or whatever. That's a normal process. It needs to happen for our body to be healthy. But it turns out the genes that control that normal cell division process have the potential, when they are mutated, to cause abnormal cell division. Too much division eventually results in the formation of a tumor, and that is the first step towards formation of cancer. So the same genes that are involved in normal cell division behavior can also, when inappropriately activated, lead to the formation of cancer. These genes are called oncogenes. So that discovery has been very important, and when we look at all of the different kinds of cancers that are so far known to exist, brain cancer, lung cancer, liver cancer, skin cancer, and so on, we find that over and over again, it's the same small number of genes that are activated by mutations that cause these cancers to form. The exciting news is that now that we know where to look, we can develop and have developed the technology that allows us to detect those mutations earlier, more accurately, and more sensitively than ever before. And in addition, that we know the targets for those mutations, we can also begin to devise therapies that allow us to reverse those mutations or to prevent their occurrence in the first place.
It's kind of scary to think that we have oncogenes in our DNA.
What stops oncogenes from causing cancer in normal cells?
Let's use these chromosome models to demonstrate. As you know and said, we have oncogenes. We also have regulator genes.
Well, what do regulator genes do?
Well, they act as switches. They turn genes on and off. And normally the regulator gene maintains the oncogene in the off state. If for some reason, through a mutation perhaps, the regulator gene becomes separated from the oncogene, then the cell could develop cancer.
Do other genes have regulators?
Well, they do. The genes that control the production of protein are called structural genes. They have regulator genes as well. And those regulator genes turn the structural genes on and off depending on whether the cell needs protein. On when the cell needs protein and off when enough protein has been produced. Now there is a structural gene that controls cell division. What do you suppose happens when that regulator is separated from the gene?
Well, uncontrolled cell division and cancer.
That's right. Some of the factors that cause gene or point mutations also cause chromosomal breakage.
So chemicals like food additives and pesticides or radiation and X-rays cause chromosomal breakage?
And that's another kind of mutation, isn't it?
That's right, because it changes the sequence of DNA, and that's the definition of a mutation.
Are there any other factors that can cause changes in DNA other than mutations?
There are. They're called viruses. Viruses are parasitic mobile genes. They cannot reproduce unless they infect a host cell. Each viral type has either DNA or RNA as its genetic material. This genetic material is enclosed inside a protein coat. The virus binds to receptor proteins on the host cell. In the RNA varieties, such as HIV, the RNA is reverse transcripted. This means transcriptase produces a DNA segment coded by the original RNA. The DNA is then transported to the nucleus and is spliced onto the DNA of the host cell. This effectively produces a mutation in the host cell's DNA. This DNA can remain dormant for some period of time before the gene for viral production turns on and begins producing new viruses. In the case of DNA viruses, the DNA crosses the cell's nuclear envelope and uses the cell's resources to replicate and to transcribe its own RNA. The host cell immediately becomes a virus factory. Virus can transport portions of host cell DNA to other host cells in a process called transduction that can also lead to genetic mutations. When viruses leave an infected cell, they occasionally carry fragments of the host cell's DNA. When they infect another cell, they may not have enough of their own DNA to fully infect that cell. Portions of the previous host's DNA may be spliced to the new host's DNA in error. The end result is a mutated cell that has not been infected by the virus and is free to replicate itself. We can now insert genes into viral DNA or RNA and use the viruses as vectors to carry the genes into specific host cells. This is part of what is referred to as genetic engineering. Mutations provide a source of variability in organisms. Now, this variability may lie in biochemical, physiological, anatomical, even behavioral differences. All genetically based differences among organisms originated at some point in the past with a germinal mutation. Now, some mutations are detrimental when they alter systems or functions that are well adapted to an organism's environment. But environments change, and so a mutation which was originally detrimental may be beneficial in adapting to the new environment. Genetic researchers are continually working to unravel the mysteries that are encoded in our genes and the influence of mutations upon them.